Listen, not to me, but to the silence underneath everything. The soft hum of the room, the steady thump of your heart. Most people think silence feels safe, like nothing's wrong. That's not really true. Silence isn't empty. It's a thin veil, barely hiding the wild chaos working underneath reality. We like to think life's solid, built from tough DNA, guarded by evolution's high walls. We tell ourselves that to kill us, something has to come hunting. A predator with claws, a virus with a purpose, maybe a hungry bacterium. But what if the real threat doesn't need hunger, or DNA, or even intent? What if you're in the way your mind dissolves, your brain turns liquid, starts? Not with an invader, but with a shape. Just a tiny, perfect error, a glitch in the way your body builds itself. This is Protocol 99, the architecture of biological entropy. Let's talk about the origami of death. To really get how terrifying a prion is, you need to let go of everything you think you know about infection. Forget the zombie deer headlines from 2015. That was a cartoon. This is older and colder. This is a law of physics, not a story about monsters. Look at the protein, the cellular prion protein, PRPC. It's inside you right now, anchored to your neurons in your brain and spinal cord. It's elegant. If you could shrink down, past your skin and deeper still, you'd see it, a ribbon, all coiled up in alpha-helices. Beautiful little springs moving through the warm, wet world inside your cells. They're flexible, they work, they're the software of life running without a hitch. These coils hum with the resonance of a healthy system. They're held together by weak bonds, just enough energy to let them fold, unfold, and get recycled by your body's enzymes. But here's the catch. There's a trapdoor in the code. Physicists talk about energy landscapes. Picture a mountain range. The normal protein, the alpha-helix, rests in a comfortable valley. Stable, but not the most stable. Farther down, deep in a dark canyon, there's another shape. That's the absolute bottom, the thermodynamic black hole. That's the beta sheet, the PRPSC. The nightmare starts with one event, the snap. Nobody really knows what lights the fuse. Maybe a cosmic ray zaps a bond. Maybe it's just thermal noise. Or, most chilling, maybe it's the touch of another broken protein. But the result is always the same. The beautiful protein loses its shape. It uncoils, it straightens, and then, like a bear trap, it slams shut into a flat, stiff sheet. This isn't biology anymore. It's a rendering glitch. As if your body's simulation just ran out of memory and swapped a detailed curve for a jagged line. Now, the new shape is flat, rigid, and stickier than anything. Normally, proteins drift past each other like ships in the night. But these glitch sheets, they want to line up. They crave order. They stack, one on top of another, like printer paper getting jammed. That stacking is called the Steric Zipper. Picture a zipper on a winter coat, the teeth snapping together, perfectly aligned. When prion sheets stack, the side chains of their amino acids mesh just like that. Here's the part that should make your skin crawl. This bond is bone dry. Life is water. Every reaction that keeps you alive happens in a soup of H2O. But the Steric Zipper is so precise, so tight, it squeezes out every last water molecule. It creates a vacuum seal, a dry zone in the middle of your wet cell. Why does that matter? Because your body's defenses, the protease enzymes, the cell's garbage crew, need water to work. They need it to chop up proteins to recycle them. But once the prion forms this watertight zipper, the enzymes can't get in. They bounce off. They can't cut what they can't touch. The prion turns into a crystal. It's not really biology anymore. It's a mineral. It's got the strength of silk, the hardness of steel. It's like a microscopic shard of glass, growing right in the soft gray folds of your brain. And this is where the domino effect kicks in. Seeded nucleation. The crystal doesn't need to copy itself like a virus. It doesn't read DNA or follow any instructions. All it takes is a single touch. A healthy, flexible alpha-helix protein bumps into a stiff, misfolded beta sheet. And just like that, the healthy one snaps. It changes shape. It gets dragged down into a lower energy state, bullied into joining the stack. 1 turns into 2, 2 into 4, 4 into a fibril. The fibril grows into a plaque. Plaques pile up until they're basically gravestones for neurons. This is what prions do. They're not alive, but their shape alone convinces living matter to die. It's an argument so powerful your biology folds immediately. There's no going back. You can't unbake a cake. Think of the beta sheet as the finished cake, the alpha-helix as the batter. Once that collapse happens, once the heat's on, the change is permanent. To fix prion disease, you'd need to break the second law of thermodynamics. You'd have to shove the boulder back up the hill, and the universe really, really hates that. You might think, fine, we'll just kill it. We have fire, bleach, radiation, we can handle this. That's 20th century thinking. Pasteur's playbook. If you boil it, it dies, right? But you can't kill something that was never alive in the first place. So, the autoclave paradox. Every hospital trusts the autoclave for sterilization, a big steel chamber, 121 degrees Celsius at 15 psi. Bacteria get ripped apart, viruses unravel, fungi turn to soup. 121 degrees. Prions, they sit there, unfazed, locked in their Steric Zipper, thumbing their nose at what we call sterile. To even stand a chance at deactivating prions, you have to crank it up to 134 degrees Celsius, soak everything in caustic sodium hydroxide, and keep it there for 18 minutes. And even then, you're gambling. There's no guarantee. It's a dice roll. The real nightmare is dry heat. Paper burns at 233 degrees Celsius. Aluminum melts at 660. But prions? Studies on sheep scabies show that even after blasting tissue at 600 degrees Celsius, the leftover ash can still infect. Just picture it. You toss an infected deer brain into an incinerator, burn it down to nothing but gray, wispy ash. You think it's gone, but the pattern remains. The template survives in the ash. Scatter it on a field, and if a healthy animal breathes it in or eats grass sprinkled with it, the whole cycle starts over. The fire that's supposed to cleanse might actually spread the curse. We're dealing with something that survives temperatures that melt metal. Prions live on the border between biology and geology. Now, the soil, the cursed ground. Chronic wasting disease sticks around because of something called clay binding, specifically montmorillonite clay. It's everywhere: your backyard, playground dust, forest mud. Prions cling to this clay. It's almost magnetic. When an infected deer dies in the woods, stumbling, drooling, shedding prions in every drop of urine and feces, finally collapsing, its body breaks down. Crows, coyotes, worms come to feed. Rain washes what's left into the ground. To your eyes, the meadow looks untouched. Flowers bloom, grass grows, but down below, in the micro-world between soil grains, the prions have found their clay. When a prion latches onto montmorillonite, two things happen. First, the clay shields it from sunlight, weather, hungry soil bacteria. The biological clock pauses. The prion becomes a time capsule. We know they can stay infectious in topsoil for decades, not days, not months, decades. Second, and here's the nasty twist, clay makes prions more infectious, not less. Studies show prions bound to clay are up to 700 times more infectious than those drifting free. The clay concentrates them, becomes a delivery system like a microscopic pill. Picture a deer grazing in a British Columbia forest. It tugs up a clump of grass, roots and all. Hidden in the dirt is a clay particle. On that clay is the ghost of a deer that died 10 years ago. The new deer swallows it. The clay shields the prion from stomach acid, smuggling it safely down into the gut. The land itself fights back. You can't shoot the dirt or cull the soil. Once a place is contaminated, it stays dangerous, like a permanent biological minefield. That's why what's happening in Florida and British Columbia isn't just some number on a chart. It's a disaster. The map keeps turning red. Florida, especially homes in Washington counties, is overrun. They tried to contain it. Didn't work. Whatever line was supposed to protect the South is gone. And up in British Columbia's Kootenay region, you can feel the panic. Officials are ordering mass culls, killing thousands of healthy deer to build buffer zones. They're destroying animals to save them from something even worse. But if the soil's already contaminated, those culls are just for show. The real story's already set in motion. So how does this thing move? How does a lifeless crystal, a gritty bit of dirt, travel from a stomach to a brain? Prions are sneaky. They're like ninja pathogen. It all starts in the gut. Maybe a deer eats it. Maybe, one day, a human does. The prion drops down to the small intestine, heading straight for the Peyer's patches basically, the gut's security checkpoints. These spots are lined with M cells, whose job is to sample whatever's in your gut and show the interesting stuff to your immune system. The M cell grabs the prion, not realizing it's making a mistake. It basically hands the prion off to the immune system soldiers, the macrophages and dendritic cells, saying, "Hey, check this out." Macrophages are the tanks of your immune system, built to gobble up invaders and dissolve them in acid. So the prion gets swallowed and dunked in a bath of lysosomes. But this thing is like a diamond. The acid does nothing. Its structure is just too tough. Now the macrophage is stuck. It swallowed something it can't break down. So it does what it's programmed to do. Carries the prion to the lymph nodes for inspection. The prion just hitches a ride, safe inside the very cells meant to destroy it, heading off to the lymph nodes, spleen, and tonsils. In the lymph nodes, it finds follicular dendritic cells and starts to multiply, using them as its lab bench. 1 becomes 2, 2 become 4. The host feels nothing, no fever, no pain, no clue that a war has started. The body's blind because the enemy looks just like its own cells. Next comes the climb. The prions need to reach the brain, so they find the nerves serving the lymph nodes, the splanchnic and vagus nerves, and start to climb. That's retrograde axonal transport. The prions literally crawl up the nerve fibers, working their way backward against the flow of nerve signals. They inch up the spine, up the vagus nerve, into the brainstem. It's a slow, relentless ascent, year after year, rung by rung, almost silent. In deer, it can take 2 years. In humans, 50. You could eat a contaminated burger as a kid, and the glitch might not reach your brain until you're old. It waits. It's patient. It almost feels like it is time itself. Now, about the species barrier. People say it's just a deer disease. Doesn't affect humans. We said the same about mad cow disease once. We were wrong. That barrier isn't a wall. It's a filter. And in 2025, the data shows the filter is starting to fail. Look at the macaque studies. Cynomolgus macaques primates, close relatives of ours. The Canadian Food Inspection Agency ran a long study, feeding them meat infected with CWD. The results were unsettling. Some monkeys got sick, but not how anyone expected. They didn't waste away or stumble. Instead, they showed strange symptoms: anxiety, tremors, trouble with fine movements. It looked like Parkinson's, early dementia, or just stress. When they tested these monkeys with RT-Quick, a way to spot prion activity, they found it. The prions were there, but something about the disease was different. If CWD jumps to humans, don't expect to see wasting or zombies. It won't look like that. We'll see a wave of anxious, trembling patients with strange kinds of brain degeneration. Who knows, maybe we're already living through the incubation period. The so-called species barrier might just be a symptom barrier. Same glitch, just a different error message on our screen. Things get even darker when you look at Scandinavia. In Norway, Sweden, and Finland, they're not just finding prions in the brains of moose and reindeer, they're showing up in the muscle too. The meat, the stuff people eat. It's almost a repeat of the BSE crisis from the 90s. When prions are in the muscle, the risk to predators and to us jumps way up. The old sporadic theory that these are just random flukes in old animals doesn't hold water anymore. The front is moving closer. So what does that mean for us? It means we're staring down the reality of biological entropy. Prions remind us that nothing is permanent. All the complicated things that make you you, your memories, your personality, your dreams, the way you love, are tied together by fragile forces that can come undone because of one twisted protein. Take fatal familial insomnia, FFI. It's a prion disease caused by a single typo in your DNA. One wrong letter and your brain's switchboard, the thalamus, just crumbles. People with FFI lose the ability to sleep forever. They drift into a nightmare of hallucinations and panic. They can't pass out. In the end, they die simply because they can't stop being awake. That's the real terror of this glitch. It doesn't just kill you. It takes apart the thing that lets you exist in the first place. It peels away the user interface of being human, until all that's left is noise. We're watching a thermodynamic mistake unfold, from Florida's soil to the forests of Norway, from deer to monkeys, from Alpha-Helix to beta sheep. Nothing saves us. Not fire, not soil, not any barrier. So we're left with just one question. How much time do we have? The clock's running, but we can't hear it. There's a crystal growing somewhere, but we can't see it. Look at your hand. Move your fingers. The signals racing down your nerves are on the same roads prions used to climb. Your body is wide open, and the hitchhiker's in no rush. This is Protocol 99. The system's breaking